Hello everyone. So this is Rafi. I am working in Aricha College of Pharmacy in the Department of Pharmaceutical Chemistry. So today I am going to introduce you a topic that is antibiotics. So this is a general structure of antibiotic. Okay. So in antibiotics, primarily we are going to discuss about the penicillins. So at first time, so it is discovered by Fleming from the fungal colony in 1982. Okay. So primarily the penicillins, they are discovered by Fleming in 92 of a fungal colony. So it is proven that they are non-toxic. So in the next at 1938, two scientists, they are isolated the antibiotic and they are Flore and Chain. Okay, so in 1928 they are discovered and they are proven that they are a non-toxic. In 1938, Flore and Chain, they are two scientists, they are isolated a pure form of antibiotics. And the first successful clinical trial done at 1941. Okay, so they are not going for any clinical trials, but at 1941 they are successfully produces a clinical trials. And that, so in 1944, so actually antibiotics, they are obtained in very less quantities in micro mLs. So this much of quantity is not sufficient to satisfy all the needs. So to recover this problem, they are, with the help of a fermentation process, they are producing in larger quantities during 1944. So similarly, its structure can be established in 1945 with the help of X-ray crystallographic method. So with the help of X-ray crystallographic method, we can able to identify the skeletal structure of the molecule. So with the help of X-ray crystallography in 1945, they are established the structure of a penicillin. Next, the full synthesis developed at Sheehan in 1957. Okay, so at 1944, with the help of a fermentation process, they are synthesizing a bulk quantity of the antibiotics, but the synthetic method is not developed. So during 1957, a synthetic method is developed to produce a larger quantities, and to produce it in the with the help of laboratory methods, they are producing a large quantities of penicillins in 1957. And similarly, isolation of 6-APA by Beckmans in 1958 and 16. So similarly, the development of semi-synthetic penicillins, so discovery of clavulanic acid and beta-lactam inhibitors. So these two are synthesized with the help of a semi-synthetic methods in the laboratories. Okay, so when coming to the definition, so an antibiotic is a substance which can able to inhibit or kill the microorganism and without producing any toxic effect to the, So an antibiotic is a substance that produced by the microorganism which has the capacity of inhibiting the growth and even destroying the other microorganisms. They are inhibit the growth or destroying the microorganisms. And when coming to the classification of an antibiotics, so there are different types of classification is there. So beta-lactam antibiotics, so there are 8 main types of antibiotics are there. The first one is beta-lactam antibiotics. Examples: penicillins, cephalosporins, monobactams and carbapenems. They are all beta-lactam antibiotics. So why they are called a beta-lactam means which having a beta-lactam ring in their structure. Okay, so all of these having a beta-lactam ring in their structure. So this is called a 5-membered beta-lactam ring. So that's why they are called as beta-lactam antibiotics. Examples are penicillins, cephalosporins, monobactams and carbapenems. When next one is beta-lactamase inhibitors. So generally beta-lactamase, it is an enzyme that is secreted by the microorganism. So that beta-lactamase enzyme, it break down the beta-lactam ring and make it inactive. So because of the beta lactamase, the antibiotics, they are inactivated and they are not showing any pharmacological effect. So that's why we are using a beta lactamase inhibitors. Those are clavulanic acid, sulbactam and the tazobactam. Next one is the tetracyclines. Examples: oxytetracycline, doxycycline, etc. Tetracyclines, they are one class of antibiotics. The next one is nitrobenzene derivatives like chloramphenicol. So these are a synthetic laboratory synthesized molecules. Next one is aminoglycosides. So primarily they are having an amino group that is linked to a glycoside molecule. So that's why they are called aminoglycosides like streptomycin, gentamicin, amikacin and neomycin. So these are aminoglycosides which are binding with a 5-membered glucose molecule. So that's why they are called aminoglycosidal antibiotics. Okay. So they are having a glucose structure in the molecule. So that's why they are called aminoglycosides antibiotics. Next one, macrolides. So when compared to structures, they are very big structures, means larger molecules. That's why they are called a macrolide antibiotics. Examples: erythromycin, roxithromycin, clarithromycin and azithromycin. So these are all high molecular weight molecules. That's why they are called macrolides. Next one is lincosamides. Okay, antibiotics, those are lincomycin and kantamycin. Next, polypeptide antibiotics like polymyxin B, clostine, bacitracin and tyrosine. These are polypeptide antibiotics, means they are having a polypeptide molecules in the structure. Okay, first of all we are going to discuss about the structure of penicillins. Okay, so when coming to the penicillins, they are having very broad classification like acid resistant, penicillinase resistance and extended spectrum penicillins. So in that 3 other classes are there, and the 4th one is beta lactamase inhibitors. So when coming to the acid resistant alternative to penicillin G, so in general the penicillin G, it is a penicillin but it is an acid resistance. It is an acid sensitive, means when we are taking the penicillin through oral route, when it is entered into the stomach, the penicillin G, when it is contact with the stomach hydrochloric acid, it is get degraded. Okay, so this is called a penicillin G. So to avoid this problem, they are producing a new type of penicillin. Those are called acid resistant penicillins, that is phenoxymethyl penicillin, that is penicillin V. Okay, so generally penicillin G, it is acid sensitive. To avoid this problem, they are synthesizing a new molecule that is called a phenoxymethyl penicillin, the penicillin V. So penicillin V, it is acid resistant. Okay, next, penicillinase resistant penicillins. So we already discussed that penicillinase, it is an enzyme that is produced by the microorganism and that will break down the 5-member lactone ring, so thereby it inhibits the pharmacological activity of an antibiotic. So to avoid this problem, they are producing penicillinase resistant penicillins like methicillin and cloxacillin. So methicillin and cloxacillin, even though penicillinase enzyme secreted by the microorganism, they are not inactivated. They shows its particular pharmacological activity. Next one, extended spectrum penicillin. So there are 3 other classes in this extended spectrum penicillins. The first one is aminopenicillins. Examples: ampicillin, bacampicillin, amoxicillin, and carboxypenicillins. Those are carbanicillin and ticaricillin. Uridopenicillins like piperacillin and mejaclocillin. And the last one is beta lactamase inhibitors. Okay, so clavulanic acid, sulbactam and tazobactam. So you have to know the difference between the beta lactamase inhibitors and penicillinase resistant. Penicillinase is another enzyme and the beta lactamase is another enzyme. So moving to the next slide. So now this is the general structure of a penicillin. So penicillin, it contains mainly the beta-lactam ring, a 5-membered beta-lactam ring. A 5-membered beta-lactam ring and an acetamido functional group. Acetamido functional group and a thiazole ring and a carboxylic acid functional group. Okay, so carboxylic acid functional group. So this is the general structure of penicillin. In that it is having a beta-lactam ring, a 5-membered beta-lactam ring and acetamido functional group and a thiazolin ring. So here as per the heterocyclic nomenclature, S means we are calling it thia and nitrogen is the aja and ol means a 5-membered ring. Okay, thiazole ring and a carboxylic acid ring. So in that, in the acetyl group, so one of the side group that is called R. So R means in the place of R you can add any of the functional groups or any of the other molecules. For example, when coming to the ampicillin, in the place of R, if you are adding an amido methylamine functional group, and amido C double bond NH is the amide functional group and this one is a methylamine, amido methylamine aromatic amino methyl and amide functional group, it is called ampicillin. The molecule is called ampicillin. Similarly, in the case of amoxicillin trihydrate, if you are adding this phenoxy group, okay, C6H5OH is the phenoxy group, phenoxy amido group, it is called amoxicillin. When coming to the cyclacillin, so in that we are going to add aniline and an amide functional group, aromatic amine with the amide functional group, it is called cyclacillin. And when coming to the cloxacillin, one more molecule, that is cloxacillin. In the cloxacillin we are going to introduce an oxazole functional group in the place of R. Okay, so in the place of R we are going to introduce an oxazole functional group. So as per the heterocyclic nomenclature, oxygen, for oxygen we are giving oxa and for nitrogen we are giving aja, and the 5-membered ring that is called ol. So this is oxazole ring. So we are going to introduce an oxazole ring in the place of R, that is called cloxacillin sodium. So cloxacillin sodium means it is a salt form. Next one, methicillin sodium. So we already discussed that methicillin, it is an acid resistance. And coming to the methicillin, 2 methoxy groups are present in the 2 methoxy groups are attached to the 6-membered aromatic ring. So in the place of R, if you are substituting with this 6-membered 2 methoxy group aromatic ring, it will give you methicillin sodium. So this is the classification of penicillins. Okay, these are also the same examples. Okay, so next one, when coming to the mechanism of action, so penicillins, they inhibit bacterial enzyme called transpeptidase. So you should have to remember this point, the transpeptidase, the main mechanism of the penicillins, those are inhibit the bacterial enzyme, that is transpeptidase. Okay, so what is the function of the transpeptidase means? This is an enzyme which is involved in the synthesis of bacterial cell wall. Okay, so transpeptidase, it is a bacterial enzyme that is involved in the synthesis of bacterial cell wall. Okay, means if you are block this transpeptidase enzyme, means the cell wall synthesis get to be inhibited. So this is the main mechanism. Now the beta-lactam ring. The beta-lactam ring is involved in the mechanism of inhibition. So this is the structure of a penicillin. In the penicillin whole molecule, the beta-lactam ring, it is involved in the inhibition of the enzyme transpeptidase. Okay, so penicillins become covalently linked to the enzyme's active site, leading to irreversible inhibition. Okay, so transpeptidase, it is an important bacterial enzyme that is essential for the synthesis of bacterial cell wall. Okay, now our drug, it will bind in our drug the beta-lactam ring. Okay, this is the 5-membered beta-lactam ring. The beta-lactam ring, it will bind to the active site of the enzyme and it will cause inhibition. So penicillin that covalently linked to the enzyme's active site, so leading to the irreversible inhibition. So this is the brief mechanism. So look at this. So this is the enzyme, this is the bacterial enzyme. Okay, so this one is the bacterial enzyme. So the beta-lactam ring, it will covalently bind to the active site, of the enzyme. So look at this, the enzyme that is binded to the beta-lactam ring. So primarily the beta-lactam ring, it contains C double bond O, means an amide functional group, CO. So C double bond O means one of the bond is pi bond and one is sigma bond. So pi bond electrons, they are shifted to the oxygen because oxygen is more electronegative when compared to carbon. So the pi bond electrons, they are shifted to oxygen. So electron density on the oxygen get increases. So we are giving a negative charge to the oxygen. Okay. Next, the carbon, it got the positive charge. Okay. So to the positive charge means it is electron deficient. To this electron deficient, the nucleus of the bacterial enzyme, it donates its electrons and form an enzyme and the carbon bond. So this is the enzyme and nucleus carbon bond. So what happened in the next step is the electron density is increases. So those electrons are again taken back by the carbon. So to maintain the balance, electronic balance, the bond between the nitrogen and carbon, it get opened. Okay, so like that the beta-lactam ring undergoes a fusion reaction. Okay, so whenever the excess of electrons, they are again back to the carbon, to maintain the valencies, electronic balance, the bond between the carbon and nitrogen, it get broken down. So thereby beta-lactam ring get break down. But the bond between the enzyme and beta-lactam ring, it is irreversible. So thereby what happened? The penicillin it blocks the bacterial enzyme. So thereby what happened? The bacterial cell wall synthesis get be inhibited, so which leads to a leakage of intracellular elements and some important intracellular enzymes which lead to the cell death. Okay, so this is a brief mechanism. So transpeptidase, it is an enzyme that is essential for the formation of bond between the L-lysine and L-lysine and the L-lysine. So for the formation of a bond between the lysine and lysine, the transpeptidase enzyme is very essential. Okay, these are the transpeptidase enzymes. But our drug beta-lactamase, our drug penicillin, what it does? It blocks the formation of a bond between the lysine and lysine. So thereby the cell wall strength get be inhibited. Okay, so this is how the drug penicillin, it blocks the transpeptidase. So this is the alanine and this is the glycine. So there have to form the bond between glycine and alanine, but our drug enzymatically it will bind to the glycine and alanine and inhibit the bond formation. So this is the cross link. Okay, so this cross link, it will helpful for the maintenance of cell wall strength. Okay, so but our drug, it covalently bind to this D-alanine and inhibit the formation of this cross linking. So thereby the strength will be inhibited. So for the formation of this cross bridges, transpeptidase is very essential. Okay, but the penicillin, it will block this transpeptidase. So thereby what happened? The cross links will not be formed. So whenever the cross links are inhibited, cell wall it loses its strength and causing the leakage of intracellular elements. So look at this, penicillins inhibit the final cross linking stage of the cell wall synthesis. Whenever the cross links are inhibited, so cell wall strength get be inhibited. And next, it reacts with the transpeptidase enzyme to form an irreversible covalent bond. Next, inhibition of transpeptidase leading to the weakened cell wall. Okay, so whenever our drug penicillin, it inhibit the transpeptidase, the cross linking between the amino acids get inhibited, so thereby which leads to the weakened cell wall. Next, cell swell due to the water entering the cell, then burst the lysis. It causes the lysis. So whenever the cell wall get thickened, then what happens? The extracellular fluid which is entered into the bacterial cell and causes cell swelling and causes the lysis of the bacterial cell. Next, penicillins possibly act as an analog of the L-alanine gamma D glucose portions of the pentapeptide. Okay, pentapeptide. However, the carboxylate group that essential to penicillin's activity. So the carboxylate group. So coming to the structure of the molecule. Okay, coming to the structure of the molecule. So this carboxylic acid functional group, that is essential. Look at this. Here the carboxylate group that essential to penicillin activity is not present in this portion. Okay, but the carboxylic group is essential, and the beta lactam ring is that is essential for the main activity of the penicillins. So up to this is today's our class. So in the next class we will discuss on the next topic. Thank you.